So hello everyone, this is MEENU AGRAWAL. Now welcome you, all of you. Now we are going to start with endomembranous system part 2. Now in the previous video you have seen about the structure of the nucleus, isn't it? Now this is what I have projected in front of you, which is giving you a 3D picture of your cell, isn't it? Now you can see very clearly in this diagram that how you can see now various structures they are connected to one another. Like you can see here, whatever you have read about the nucleus, complete structure is here, and here you can see the nucleus is there, and you can see endomembranous system, how it's connected with the outer membrane of your nuclear membrane. And you can see here that no, some membranes are of this are not having any ribosomes on it. They are your smooth endoplasmic reticulum. As you know that endoplasmic reticulum is of two types, we will come to that. Okay, and now you can see here that these are your Golgi bodies, and how these are your what vesicles. These are transport vesicles, how they are transporting substances from one membrane to another membrane, and how they are moving the substances out of the membrane. Okay, so the structure is quite clearly visible. Now the question arises here is that okay, find the, the nucleus has got what? It has got DNA, and that day also I told you that DNA has got what? It has got an information. Okay, as I told you that this controls your entire activities of your cell, isn't it? Now if it has got the information which you say, okay, so if it has the information, and information for what? For controlling the various activities of the cell, then this information must express itself, isn't it? How does it expresses? Okay, so suppose you have an information. Now what do you do with that information? You do something with that information. Just having that information is of no use. Suppose you know about your subject, now ultimately you have to write in your exams, so somewhere it has to come. You have to put into a response form, into an action form. Okay, so similarly in your body, what do you find? DNA has got this information, fine. Now what it does, it now passes that information. Okay, so it passes that information into the form of what? Messenger RNA, mRNA we call it. Okay, and this process is called as transcription. You will read in higher classes about it. Then this messenger RNA, it moves out from these nuclear pore. Okay, it moves out from this nuclear pore, and this transcription takes place where? In the nucleus. Okay, it takes place where? In the nucleus. And now from the nucleus it moves out in the form of messenger RNA and comes into the cytoplasm. Now what happens in the cytoplasm? You know that the protein is formed. Protein synthesis takes place. Okay, and this process is known as translation. Okay, now you know that this protein is being synthesized. You have read also about it by ribosomes. So this messenger RNA has got a message which is read by ribosomes, and what is happening? Your proteins are being formed. Okay, according to the message, amino acids are added. Okay, and your complete protein is being formed. Now these ribosomes, now you know that the ribosomes are what? There are cytosolic ribosomes, as you can see here in the diagram also. That see, here in the diagram you can see these are the ribosomes which are lying in the cytoplasm, and there are ribosomes which are also lying on the membrane, on the cytosolic surface of the membrane. Now the question rises, how does your this messenger RNA and this ribosomes know that whether they are to be synthesized in the cytoplasm, whether they are cytosolic proteins, or whether they are to be synthesized in the endoplasmic reticulum. Okay, so now moving on with that, just I will take a. Now you can see here what is happening is that now the messenger RNA, this mRNA molecule, it has got a sequence. Okay, it has got what? A sequence which we call as a signal. It has got a signal sequence. Okay, and that sequence can be read by your ribosomes. Now, if they read that it is, like suppose I just draw for you something like there is a pool of what? Ribosomes, okay, in the cytoplasm. Now, this messenger RNA comes. This messenger RNA comes, okay, and what happens? Your ribosomes attach to it and starts forming. Now you call this structure as what? You call it as polyribosome. Okay, and it starts synthesizing, and this is happening in your cytoplasm. And another messenger RNA comes, and another messenger RNA comes, and what happens? It attaches itself. It attaches itself to the membrane. Okay, it attaches itself to the. Suppose this is the membrane, endoplasmic reticulum. So it attaches itself. The ribosomes brings it, and what happens? It attaches it to the endoplasmic reticulum. It attaches it to the endoplasmic reticulum. Okay, why? How it attaches? This is the question, isn't it? This is something very common coming to the mind. Okay, fine, your now messenger RNA is being formed by the nucleus. Then how does it bifurcate? Which has to be formed in the cytoplasm and which has to be formed in the endoplasmic reticulum. So based on that signal sequence, they do that. And remember one thing, that those proteins which needs to be glycosylated, this glycosylation cannot be done in the cytoplasm. Okay, because the environment is highly reducing in the cytoplasm. Okay, therefore what is happening, for glycosylation, even if they are formed in the cytoplasm and they need to be glycosylated, they have to be transported to the endoplasmic reticulum. Okay, so what do we find? That even your even the proteins which are formed in the cytoplasm, they also move to the endoplasmic reticulum, which has to be glycosylated. Okay, or you can say which has to be which has to be secreted out or transported out. Okay, which needs to be transported or secreted. Okay, so they have to move into the this endoplasmic reticulum. Then only this membranous structure will cover it in a vesicle like structure, in a bag like structure, and help it in transporting from one place to another place. Okay, so this is happening in your way, in your endoplasmic reticulum. Okay, so let's look on basic things. Like that there are what? There are two types of endoplasmic reticulum. If I write endoplasmic reticulum, it is of what? It is of two types. What are they? They are your smooth endoplasmic reticulum and your rough endoplasmic reticulum. Okay, so rough, why? Because of attachment of ribosomes, the appearance is rough. It's in now what are the different functions of the smooth endoplasmic reticulum? Let's look into it. See, smooth endoplasmic reticulum is basically involved in the formation of your what? In your lipids. Okay, and you know about lipids. Okay, a little bit you know about lipids, that they are what? They have got a hydrophobic nature. So most of the lipid formation is within the membrane. It's not towards the cytosolic side. Okay, only those lipids can be formed which are made up of glycerol and all. Okay, which are water, have got the oil component in them. Okay, so they have got a hydrophilic end also. Now, apart from lipid formation, smooth endoplasmic reticulum has got a major role in what? The carbohydrate metabolism. Okay, they have a major role in your what? Detoxification. Now, what is detoxification? Detoxification is a process where what you do? Where now something very harmful is converted into less harmful. Okay, so very harmful substance is converted into less harmful substance by detoxification. So various drugs, alcohol, everything now is even your ammonia, which is formed from by your amino acids, it also get detoxified into urea, into your liver. So various drugs, all these things are getting detoxified. Okay, and you know that your liver cells have got a huge number of what now the smooth endoplasmic reticulum. Now coming to what, they also act as a storage site for calcium. When you will come to muscles and all, you will study about them, that these Golgi bodies, they store, smooth endoplasmic reticulum store a huge amount of calcium in it. Okay, so this is about smooth endoplasmic reticulum. Now, if you look at the structure of rough endoplasmic reticulum, so you know it is involved in the formation of various kinds of proteins. Even your, there is a term which is given, you must have come across that term, membrane biogenesis. Now, since you can see by endoplasmic reticulum, what is happening? Lipids are also being formed and proteins are also being formed, which are the component of your membrane. So constantly when something is being moving out, so those vesicles are fusing with your membrane and they are becoming one of the component of your membrane. Okay, so this is about that. And let's look into that, how endoplasmic reticulum, see, endoplasmic reticulum, if you look into these ribosomes, are in a very close connection with your endoplasmic reticulum. Just look at this figure which is illustrating it. Now you can see here that what is happening here. You can see that ribosomes are forming a very great connection with your endoplasmic reticulum, and whatever protein is being formed, they get inside into these lumen. Okay, they get inside into the lumen of your endoplasmic reticulum, and there what is happening, sugars are added to it, they are getting glycosylated, and they are getting glycosylated, and transport vesicles deliver glycoproteins to the cis end of your, so we will come to that later. Now remember one thing, now what is happening here, we will come to Golgi bodies also. So what do we find here, that now whatever protein is being synthesized, they are getting folded. You know that protein has got a conformational structure, and if that conformational structure is active, if that structure is conformationally, it has to be conformationally active, it should be. If it is not active, then now there are a set of proteins in your endoplasmic reticulum, which we call them as, what do we call them as? We call them as chaperones. We call them as chaperones, which fold these proteins into a conformationally active structure. If not formed, okay, then those proteins are degraded. Okay, so these chaperones are able to do that. Okay, now if you come to see this diagram is very nicely, it is showing you that you can see here now that the Golgi body, now how the endomembranous systems are connected, you can see here very nicely. Now like how the proteins which is being synthesized in the endoplasmic reticulum is moving into the various stacks of the Golgi body. We will come to Golgi body. Okay, and you can see here here also I can show you, this is called as the cis end. Which end we are calling as the cis end? We are calling those ends as the cis end, which end is what? Receiving. The cis end is what? It's receiving. It's receiving proteins from your endoplasmic reticulum. Okay, and you can see that they are in a stacks of cisternae are there. And what is happening now from the cis end? You can see here from the cis end, now these gets now filled in here. So these vesicles are formed from your endoplasmic reticulum. Okay, now they come and join at the cis end. Okay, now what is happening in your Golgi body? Either it will be more glycosylation is there, or disulfide bonds are being formed, and so all those things now they are going to take place. So Golgi body has got various roles. Okay, so what are the roles of the Golgi body? Let's look into that. So we can see here that Golgi body has got various. Now we were discussing about the various roles of the Golgi body. So let's come to the various roles of the Golgi body first. Then I will explain the diagram. Golgi bodies. If you look at the Golgi body, it does various things. Now it does what? Sorting. It does what? It does modification. It makes glycolipids, it makes glycoproteins. Okay, modification is being done. Then what is done? It is also storage site, storing, and then what? Transporting now things into vesicles to their destination. It's moving from there, from the Golgi bodies, as we can see here. And now what do you think? I can give an example of Golgi body like a factory, which is receiving the raw materials like from endoplasmic reticulum, and where what is happening, like if a lace factory is there, what is it doing? आलू चिप्स, आलू आ रहा है और चिप्स बनना है, तो उसमें क्या हो रहा है? ऑल काइंड ऑफ मॉडिफिकेशन इज बीइंग डन. उसको सॉर्ट किया जा रहा है, अच्छे आलू, खराब आलू, है ना, फिर उसको कट किया जा रहा है, वेरियस थिंग्स आर बीइंग डन, मॉडिफिकेशंस आर बीइंग डन, different flavor, फिर storage, फिर packing, and then transporting. Okay, so packaging is also one of your function of your what? Your Golgi bodies. So packaging is also one of the function, okay, of your Golgi bodies. Now you can see here in the diagram the end which is receiving. This end is what? Is the cis end of your now Golgi body, and the end which is from, and you also see from moving from one stack of Golgi to another body, just you see they are being packed into what? Into your trunk, this vesicles, and they are moving from here, and then they're coming again, and then moving again, and like this, and ultimately these are your secretory vesicles which are being formed from which end? What is this end called? This end is called as trans end. Now, if you look at the enzymes, so definitely the cis end has those enzymes which are needed early, okay, and trans end enzymes contain enzymes which are needed late in the formation of the protein. Okay, now what you can see here, that these are the transport vesicles, they are carrying it to their destiny. Now, what is happening? What are the different, now you can say the fate of these transport vesicles? Either they could be what? If I talk about the fate, either they could be secreting a secretory molecule, secreting something, you can see here also there is what? There is a secretory system. This is a secretory system, and this system is what? It's what? It's an endocytic system. Okay, something is moving in from here and something is moving out from the cell. Okay, so two systems are, that is endocytosis and exosmosis. You can say exocytosis is happening here and this is endocytosis. Okay, two pathways are running. So it could be secreting something. Okay, it could be now releasing something into the extracellular fluid, to nourish it, or it may be releasing your protein which may be acting as a membrane protein, or you can say peripheral protein which you have read. Okay, fine. And it may be also diffuse and nourish. It may also act like a signal molecule for others, no, for other cells, for other cells. Okay, so it could be acting as a various molecules, okay, which is to be secreted out, you know. Now what do we find, that here there is another pathway now which you are finding key, which is, now you can see here from your Golgi body is only your this molecule, as now you know that lysosomes are formed from your now Golgi bodies. So these Golgi bodies, your these enzymes are hydrolytic and in nature. We will talk about them. So these lysosomes, then whatever comes inside, now we call that as what? Endosome. We call it as what? We call it as endosome. Okay, so this endosome is there, and then this endosome it joins with the lysosomes and forms, and then it forms ultimately your lysosomes. Okay, so this is the two pathways which we can see, and they are all endomembranous pathways. Now moving on to your lysosome. Okay, now if you look at the structure, you know that lysosomes are what? They are nothing but bag-like structures. Okay, they are what? Bag filled with what? Filled with hydrolytic enzymes. As you can see here, it contains various acid hydrolases, like nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases. So they act on these molecules and they have a major role. Now they have a major role in digestion. So they digest various substances. They digest various molecules. And then pH is how much? It is 5. And that pH is maintained. You have read also earlier. Now you can see here there is another pump. There is a pump by which hydrogen ions are being pumped into this vesicle. Okay, so via this hydrogen pump, it is being constantly pumped into this vesicle so that the pH is maintained, so that these enzymes can act upon. Okay, and if you look at the cytosolic pH, you can look at the cytosolic pH. It is what? 7.2, which is very which is basic, and this is highly acidic. Now the question arises, there are a lot of questions that must be coming to your mind, like how the membrane proteins, the bag which is holding such an acid, is their proteins are being maintained, how? So those proteins get glycosylated. Okay, so if you look at those proteins are highly glycosylated to get prevented from the action of your acids. Okay, they are highly glycosylated. Okay, and they help in what? They also help in what? Cleaning your cell. You know that, they help in cleaning of your cell. They are also involved in cleaning of your cells. as you can see in this diagram, what is happening? Just see. Now this is the your lysosome. Okay, so this is your lysosome, as you can see here. Now this lysosome, okay, what is it doing? It is able, even now, like some bacteria is moving inside the cell. Okay, so what it does, it is able to digest, and this digestion is, remember one thing, that it is highly controlled. This digestion is very highly controlled. And it is happening in a very controlled manner, okay, by these lysosomes, whether it's an extracellular digestion, something which is coming from outside, or whether it is within intracellular digestion. Even intracellular digestion is here, as you can see here, like what is happening, mitochondrion is there, and a worn out mitochondria is there, which is not working inside the cell. Okay, now this mitochondria is now fusing with your lysosomes, and it is getting what, digested. Now that's how the cleaning of the cell is being done. Worn out cells are being removed. Okay, even it is protecting you, like from bacteria. If it is fighting, it is protecting from infection, isn't it? So it is protecting in that case. And if you look at this here, like something has moved inside it, you know, and some food material, like by pinocytosis or something has moved inside, and that is called as what? What do we call it as? We call it as endosome. What do we call it as? We call it as endosome. Okay, now that endosome, it grows in size, you know, and then it fuses with the lysosome, and the lysosome digests that substance. Okay, so lysosomes are now very important organelle, you can say, in a way that it helps in cleaning the cell, in digesting the food materials, and providing it to various parts of the cell also. And see, this lysosome, if you you also know that they are called as what? Suicidal bags. When do we call them as suicidal bags? See, they are rich in hydrolytic enzymes, and in certain cases, when they are not able to now clean up the toxic materials which is being produced by the cell, or not able to clean the cell or perform its function very well, then in that cases, sometimes these lysosomes, they burst and they digest the entire cell. That's why they are called as what? We call them as suicidal baths. Okay, so like in case of, you can see, I can give you an example, like in your menstrual cycle. Okay, what is happening now? Bleeding takes place, how is it that bleeding is taking place? You even your cells slough off. Okay, so it is happening because of this now suicidal action there. Your lysosomes, they burst themselves. Okay, and where they eat their own worn out cells, then that condition is called as what? It is called as autophagy. What do we call it? Autophagy. मतलब खुद का ही चीज खा रहे हैं. That is called as autophagy. So with this, we have completed our endomembranous system, isn't it? And in the next class, we will start with the other organelles. Okay, with this, thank you. Bye bye. Keep subscribing and watching.